Continuing with our series of lectures on DNA replication, today we will discuss the other important characteristic features like semi-discontinuous nature, bidirectional nature, and the fact that primer synthesis is absolutely essential for DNA replication. Let us start with discussing whether DNA replication is discontinuous, semi-discontinuous, or continuous. This is a DNA duplex where you can see that both the strands are anti-parallel to each other. One strand is 3' to 5' and the other is 5' to 3'. If we are charged with the task of designing a DNA replicating machine, we might come up with a system similar to the one on your screen right now. DNA would unwind to create a fork, and two new DNA strands would be synthesized continuously in the same direction as the moving fork. This is the continuous model. However, this scheme has a major fatal flaw. It demands that the replicating machine be able to make DNA in both the 5' 3' and 3' 5' directions. That is because of the anti-parallel nature of the two DNA strands. If one strand runs 5' to 3' left to right, the other must run 3' to 5' left to right. But the enzyme which synthesizes DNA, the DNA polymerase of all natural replicating machines, can make DNA in only one direction, and that is 5' to 3'. That is, it inserts the 5' most nucleotide first and extends the chain towards the 3' end by adding nucleotides to the 3' end of the growing chain. Following this line of reasoning, Okazaki concluded that both strands cannot replicate continuously and hence this model cannot exist. He proposed the semi-discontinuous model where DNA polymerase could theoretically make one strand, the leading strand, continuously in the 5' 3' direction, but the other strand, the lagging strand, would have to be made discontinuously, as you can see in the figure. The third model proposed was the discontinuous model where both the leading and the lagging strand are synthesized discontinuously in the 5' 3' direction. Let us understand the semi-discontinuous model. We have a DNA duplex which opens up to form a replication fork. Both strands are being synthesized in the 5' 3' direction. The discontinuity of the synthesis of the lagging strand comes about because its direction of synthesis is opposite to the direction in which the replicating fork is moving. Therefore, as the fork opens up and exposes new regions of DNA to replicate, the lagging strand is growing in the wrong direction away from the fork. The only way to replicate this newly exposed region is to restart DNA synthesis at the fork behind the piece of DNA that has already been made. This starting and restarting of DNA synthesis occurs over and over again. This was the semi-discontinuous model proposed by Okazaki. The short pieces of DNA thus created would of course have to be joined together somehow to produce the continuous strand that is the final product of the DNA replication. Okazaki's model of semi-discontinuous replication made two predictions that his research team tested experimentally. Because at least half of the newly synthesized DNA appears first as short pieces, one ought to be able to label and catch these before they are stitched together by allowing only very short periods, that is pulses of labeling with a radioactively labeled precursor. To test this prediction, Okazaki and colleagues gave shorter and shorter pulses of labeled thymidine to E. coli cells that were replicating the T4 DNA. They grew E. coli and added thymidine in the medium for a few seconds and spun down and broke the cell to stop labeling and analyzed. To be sure of catching short pieces of DNA before they could be joined together, they even administered pulses as short as 2 seconds. Finally, they measured the approximate sizes of the newly synthesized DNAs by ultracentrifugation. Already at 2 seconds, some labeled DNA was visible in the gradient. It appeared that all of the label was in very small DNA pieces, 1000 to 2000 nucleotides long, which remained near the top of the centrifuge tube. If one eliminates the enzyme DNA ligase responsible for stitching together the short pieces of DNA, these short pieces ought to be detectable even with relatively long pulses of DNA precursor. Experimental evidence for this goes like this. They took a T4 mutant containing a defective DNA ligase gene. The small DNA fragments accumulated to very high levels when the stitching enzyme DNA ligase did not operate. This proved that the Okazaki fragments are not just an artifact of very short labeling times. 50% of the newly replicated DNA is indeed in the form of fragments. Thus, replication really appears to be semi-discontinuous. One strand replicates continuously, whereas the other strand replicates discontinuously. Let us now move on to the next characteristic feature of DNA replication, that is bidirectional replication. In the early 1960s, John Cairns labeled replicating E. coli DNA with a radioactive DNA precursor and then subjected the labeled DNA to autoradiography. The figure on your screen shows the result. The structure is called a theta structure because of its resemblance to the Greek letter theta. The schematic here shows that DNA replication begins at the origin with the creation of a bubble, a small region where the parental strands have separated and progeny DNA has been synthesized. As the bubble expands, the replicating DNA begins to take on the theta shape. Have a look at the animation here showing the replication of circular DNA by theta model to form two circular DNA duplexes. The theta structure contains two forks as can be seen in the figure. This raises an important question. Does one of the forks or both represent sites of active DNA replication? In other words, is DNA replication unidirectional with one fork moving away from the other which remains fixed at the origin of replication? Or is it bidirectional with two replicating forks moving in opposite directions away from the origin? Cairns' model was not designed to answer this question. However, Elizabeth Gurasitis and R.B. Wake showed clearly that DNA replication in the bacterium is bidirectional. This is say the Bacillus subtilis genome with its origin. These investigators' strategy was to allow Bacillus subtilis cells to grow for a short time in the presence of a weakly radioactively labeled DNA precursor and then continue growing it in the same low radioactivity pulse. The labeled precursor was thymidine. The purple color here represents the weakly labeled DNA strands produced during the low radioactivity pulse. Now, expose the cells for a short time to a strong radioactive precursor. The orange color here represents the highly labeled DNA strands produced during the later high radioactivity pulse. Because both folks picked up the high radioactivity label, both must have been functioning during the high radioactivity pulse. DNA replication in Bacillus subtilis is therefore bidirectional. Thus, this figure shows unidirectional replication and this shows bidirectional replication. This is the third important feature of DNA replication. Do all genetic systems replicate bidirectionally? Apparently not. Thus, it is not a universal feature. For example, some plasmids like Col-even plasmid replicates unidirectionally. Let us now move on to see what are the requirements for DNA synthesis. If we have a DNA template, DNA polymerase, and add the precursors, that is all the nucleotides and other small molecules needed to make DNA in an append-off, will new DNA be made? The answer is no. Why does new DNA synthesis not take place? What is missing? Well, the missing component here is a primer, a piece of nucleic acid that the polymerase can grab onto and extend by adding nucleotides to its 3' end. This is because DNA polymerase cannot start DNA synthesis de novo. Say this is a single-stranded DNA template. A short oligonucleotide is synthesized at the 5' end. This is a primer. This primer is not DNA but a short piece of RNA. This is now called a template primer junction. To the 5' end of the primer of this junction, DNA polymerase can bind and start DNA synthesis. Primer synthesis is absolutely essential for DNA replication to take place, and this is brought about by an enzyme primase. First, a replicating fork opens up. Next, short RNA primers are made, and then DNA polymerase adds deoxyribonucleotides to these primers, forming DNA as indicated in the figure by the red arrows. This is the lagging strand which is synthesized discontinuously. The leading strand is synthesized continuously. The first line of evidence supporting RNA priming was the finding that replication of M13 phage DNA by an E. coli extract is inhibited by the antibiotic rifampicin. Now this was a surprise because rifampicin inhibits E. coli RNA polymerase and not DNA polymerase. The explanation is that M13 uses the E. coli RNA polymerase to make RNA primers for its DNA synthesis. However, this is not a general phenomenon. Even E. coli does not use its own RNA polymerase for priming. It has a special enzyme for this purpose, that is the primase. Perhaps the best evidence for RNA priming was the discovery that DNAs cannot completely destroy Okazaki fragments. It leaves little pieces of RNA 10 to 12 bases long. Most of this work was carried out by Tsuneko Okazaki, Reiji Okazaki's wife and scientific colleague. She and her co-workers took the Okazaki fragments and subjected them to DNAs. They then subjected the surviving labeled primers to gel electrophoresis. The primers produced clearly visible bands that corresponded fragments with a length of 10 to 12 nucleotides. Treating these fragments with RNAs clearly demonstrated that they are RNA in nature. Thus, these RNA primers were discovered by Tsuneko Okazaki. With this, we come to an end of this lecture. In this lecture, we have seen three important features of DNA replication. In the next lecture, we will be talking about the enzymes required for DNA replication.